Thank you for that introduction. And of course, thank you to the organizers for allowing me to speak today. So I'm a graduate student at Wayne State University, and I'm going to talk to you about regulatory variation. So our lab studies regulatory variance, and one of the reasons that we find this to be such an interesting topic comes from GWAS results. As you can see here in this example from Crohn's disease, a lot of the associated variation comes within or outside of any gene body, suggesting that most of the functional variation is due to changes in regulation rather than changes in the genes themselves. However, predicting the impact of this variation on the regulatory sequences is very challenging. In order to do that, the first thing we need to know is where these regulatory sequences are, and one technique to figure this out is DNase footprinting. This is a technique that takes regions of... Oh, sure. Sorry. Is that better? Oh. So regulatory variation, we're looking to find them. So one of the ways that we find these regions of regulatory regions is using DNase footprinting, which takes these sections of open chromatin, usually associated with gene transcription, and it uses an enzyme called DNase to chop up the DNA. And if you sequence this at high enough coverage, what you see is these footprint patterns, which is where the transcription factors are binding. If you use a method such as centipede to pair a motif model with the footprint location, you can start to get at which transcription factors are binding and where. Then to actually show the effect of a sequence variant on binding, there are two types of analyses you can do. One is you can do QTL studies, which has been mentioned a couple times today, but you need a large cohort of individuals, each with the different genotypes. The other thing you can do is allele-specific analysis, where in a single individual, you just look at the reads overlapping the position that you're interested in. So in this talk, I'm going to describe how we've used DNase footprinting to generate a set of predicted regulatory variants, and how we validated them using allele-specific binding analysis, and finally, how we've tried to integrate our annotations into GWAS studies to show some downstream function of these variants. So for this study, we used over 600 samples from the ENCODE and Roadmap Epigenomics projects, as well as sequence motifs from the Transvac and JASPAR catalogs. And using this data, we applied the centipede method to determine which factors were active in which tissues. And how we do this is we take, for every motif, we do an initial scan where we look at some of the better motif matches across the genome, and we use centipede to determine the presence or absence of a footprint at that position. And so, for example, we can identify which tissues are active, or which transcription factors are active in which tissues. So in this example, this would be a factor active in LCLs, but not necessarily in HUVECs. So the next step was then to try and recalibrate some of these sequence models, mainly because a lot of the ones that are in the databases available today are very outdated and based on non-human data. And so in order to do this, we used the sequences that were present at the footprints we saw in our initial scan, as well as some human sequences that have orthologous matches to the motif in chimps and macaque. And the reason for this is we didn't want to bias our new motif models by using just the best footprints. We wanted to add some degeneracy, and we figured this is a much better way than just randomly permuting the bases. So then with these updated models, we scanned the whole genome for all of the all of the matches, and we did all of the footprinting. And so here's an example of one of the updated sequence models. This is for NRSF. The seed is on the top that we used from the database, and the bottom is our recalibrated one. You can see that the core motif doesn't really change, but the importance of the bases does. And compared to a ChIP-seq golden standard using, again, ENCODE data, we can see that our recalibrated models have a lower false discovery rate and a higher sensitivity than the seeds. And here, if you compare the centipede predicted probability of binding to the ChIP-seq occupancy, we see, most importantly, that we're capturing this range of accessibility rather than just kind of stratifying by not bound or bound. So across all of our samples, we identified about almost 1,400 motifs with a footprint in at least one sample. And on average, this is about 600 active motifs per sample. And when we overlapped these footprints with known variants in the 1,000 Genomes catalog, we were able to make a binding prediction for both alleles using the PWM match score in the centipede model. And we call these footprint SNPs. We have 5.8 million SNPs within these footprints. And then using the differential scoring, we noticed that 3.8 million are predicted to have a quantitative impact on the binding affinity by at least 20-fold. We call these effect SNPs. And then a small subset of these are predicted to switch the probability of binding from likely bound to likely not bound, and we call these switch SNPs. So then to validate these binding predictions, we went back to the DNase data and independently examined the allele counts at these SNPs. And to do so, we used the quasar method that Roger mentioned earlier today. And instead of looking at allele-specific expression, we looked at allele-specific binding. One caveat, though, is that the samples that we were using for this were collected with the intention of regulatory mapping and not allele-specific analysis, so the coverage is a little bit lower than we would have liked. And we also had to discard cancer samples and samples with no really obvious chromosomal abnormalities because those just aren't great for allele-specific analysis. After all that was said and done, though, we still had 316 samples that we were able to call ISCB on. And overall, we saw, if you look at all of the SNPs that fall within DHS regions, we see a slight, about a 4-fold enrichment for ISCB signal versus the null. And if you look at all of those that are also within annotated footprints, we see a slightly higher, about a 6-fold enrichment over the null. However, when you examine just the effect SNPs, you see a 9-fold enrichment for ISCB, and we see a 14-fold enrichment for the switch SNPs. And the fact that these annotations are enriched for ISCB further supports the hypothesis that these predicted categories are truly functional. We used the story FTR procedure to do multiple test correction, and we observed that only a small fraction of these DHS SNPs are likely to actually impact binding. Slightly more if you look at the footprint SNPs, but still most of them are going to be neutral. However, if you look at the effect SNPs, about 56% of them are predicted to impact binding. So given this large category of variants that we have, we wanted to know if there are any genomic characteristics that separate those that affect binding from those that do not. And we see that these effect SNPs are enriched for having low allele frequencies. So here I have the allele frequency binned, and you can see that the dark red bars, those that are effect SNPs, and the light red bar, those that are footprint SNPs that are not effect SNPs. And this is similar to what's been seen in coding regions. If you look at non-synonymous versus synonymous, non-synonymous SNPs have a lower minor allele frequency. And we also saw that these effect SNPs tend to be depleted in promoter regions, about within 300 bases of a TSS. And they're also more likely to be cell type specific. And what I mean by that is that they sit in the binding sites of motifs that are of factors that are only active in like 5 or fewer tissues. So then we next looked at the degree of ASB affecting these different factors. And to do that, we calculated the ASB enrichment ratio, which is just the number of ASB SNPs, or SNPs displaying ASB, versus all of the heterozygote SNPs within or across all of the motif instances. And we determined which motifs were significantly enriched or depleted compared to sort of the average that we were getting. And we saw 32 were enriched and 56 were depleted. I don't know, well, based on the coloring, so the red here indicates that they're active in fewer tissues, so this kind of recapitulates what you see for the, from the SNP data. And you can see factors like AP1 and CREB that are enriched. Whereas for the depleted factors, we see a lot of housekeeping factors like CTCF and E2F that are active in almost all of the tissues we looked at. So given these distinct features, we wanted to know if a lot of them had arisen as a result of natural selection. And this is a tricky question to get at, but we used a method based on the McDonald-Kreitzman test, where you traditionally look at protein coding regions, and we modified it to work with our motifs, considering the whole body of motif instances, and a single motif as a functional unit. And for polymorphic sites, we just classified whether a motif had an effect SNP or a footprint SNP that was not an effect SNP. For the divergent sites, we again looked at the, for all of the positions that did not have a polymorphism in humans, we looked at the predicted binding score in chimps. And if the difference between humans and chimps was the same cutoff that we were using, the 20-fold, we considered it a functional change, and if it was less than that, we considered it a silent change. And now based on, so you do it, then the rest is similar to the MK test where you generate a score, and from the score you can estimate the direction of selection. And so what we see is that the XY line is about here. So one thing we see is that most of them are shifted towards purifying selection. However, if you look at the factors that are undergoing, that have higher scores for positive selection, we see factors that tend to regulate neural and developmental processes. And so obviously there's a lot more that we'd like to do at this front, but I think it's a really nice example of how we can start to understand some of the basic biology behind these regulatory variants. So finally, I'd like to show you a little bit of work that we've done to tie these regulatory variants into a downstream mechanism using GWAS meta-analysis studies. And to do so, we've been using a model called FGWAS, which was developed by Joe Pickrell and recently published. And the gist of this is that you can use genomic annotations to try and figure out which annotations are associated with the GWAS hits. And so here's an example of where we put our footprint annotations for each motif into a height study. And so you essentially get an enrichment score for each factor based on how enriched that factor is for having associated SNPs in their binding sites. And what we see for this height example is a lot of factors consistent with the biology of height, those that are involved in development, cell proliferation, and stem cell maintenance. Similarly, we looked at an LDL study, a low-density lipoprotein study, and we see, again, biologically relevant results popping out. We see factors that are enriched for inflammatory and immune function. We see the liver-specific HNF4 alpha factor. And we see the USF-EBOX factor, which has been linked to familial hyperlipidemia. So in addition to just looking at the annotation-wide enrichments, we can also zoom in and look at the individual association blocks. And so here's an example using that USF-EBOX factor I mentioned, where so there's this large association peak, and this SNP here happens to fall within a motif for the USF-EBOX shown here. And the T allele is an effect SNP. It's associated with a decreased amount of binding, and it's also associated with an increase in LDL levels in the blood. And my colleague Cindy Kalita has been doing some reporter assay work on validating this and several other variants. And for more on that, you should see her poster. I think the evens are tomorrow. So in summary, we've annotated 5.8 million SNPs to be within active regulatory regions. However, not all of them really seem to show any effect on binding. But we do notice that about 56% of those that we predict to have an effect really do, which underscores how important it is to have a good predictive model. So then looking at these SNPs, they have low allele frequencies, and they tend to affect tissue-specific distal enhancer elements. And finally, by using these functional annotations and integrating them into GWAS studies, you can really start to identify potentially causal GWAS SNPs and the tissues and transcription factors affected, which really starts to give you the mechanism behind the associated trait. I'd like to thank my advisors, Roger Piquet-Reggi and Francesca Luca, as well as everyone in the lab, including Chris Harvey and Cindy Kalita, who really helped on this project. Our collaborator, William, at the University of Michigan, and of course, Wayne State, and our funding sources. And like Roger said, we're definitely looking for postdocs. There's a lot of really cool stuff going on in our lab. So send us an email or check out our website. Thank you.
Absolutely beautiful work. So I have two questions. The first one is, if you look at the effect direction on the DNase for a particular site, does it ever vary from tissue to tissue? In other words, you see that some regulator might actually switch from being, contributing or, towards DHS?
That is a great question. I can't think of a specific example of that off the top of my head, but because we do this type of analysis independently within each tissue sample that we look at, that is definitely something that you would see if it was a tissue-specific directionality.
The second question is, there's a lot of work, a lot of sort of thinking about whether in every LD block you have one causal variant, or whether the entire haplotype is in fact influencing the directionality of effect based on multiple causal variants, each of them affecting perhaps slightly a different regulatory element, also known as the multiple enhancer variant hypothesis. My question is, how often when you go in one of those haplotypes, basically have you looked at the relationship between all of your DS, affecting SNPs, whether you sometimes see multiple SNPs in the same haplotype, or whether when you see one, you've more or less explained away the effect of that LD block. And now if you look at neighboring variants, you actually don't see another one there.
Yeah, another good question. We have not started to formally test that, but as I've been starting to try and put our annotations into the genome browser to start to actually see what's going on, there's definitely regions where these effect SNPs tend to be clustered in regions where lots of factors are binding. And whether they're all causal or which one's causal would be a really nice question.
So you do have examples where you have multiple such SNPs in the same haplotype block?
No, I don't think so.
Not yet. Okay, great.
Yeah, we have not looked at
Wonderful work. Thank you.
Thanks.
I also really thought that was beautiful. I wanted to ask you if it's related to another axis of the question that Manolis asked. One of the big problems, of course, in connecting transcription factors with functions and cisreg targets is that there are often multiple members of a transcription factor family expressed in the same cells at the same time. And your motifs are really trained on particular members of these families, but they could be bound by other members of the families. So one class of SNPs that I would imagine that you might get would be ones that would actually make a USF EBOX a little bit more permissive for binding by some other EBOX factor. So I'm just curious, have you actually gone through your data to try to group these occupancy patterns, these motif families by family, and actually try to figure out whether there's intra-family competition going on?
Yeah, that's a great point. And it's worth noting that we have a lot of, we have many more motifs than we do factors that we're really looking at. And
ATF CREB also.
Right, yeah, exactly. Yeah. So one thing that's on the horizon is to try and cluster these motifs to really drive at sort of these functional units, kind of irrespective of what the factors are, so we can look for those families. In terms of sort of the second part of what you're saying, whether or not changing it makes it permissive to a different type of factor, that's a really interesting question that I would like to look at, but we haven't yet.
Yeah, good luck.
Okay, so let's thank Greg, Irene, and Roger again.